So if it's all working, I will start. So I start by presenting myself. I'm Ugoline Godeau from INRAE in France. And I would like to take the opportunity of this consortium to present a current sidewalk that is in progress from work package 5. So the main idea is to look at the link between the in-hive temperature and the colony strength, and especially the regulation of this in-hive temperature. And for that, I want to first present to you our ideas of analysis and the application on another data set that we want then to apply on Be Good later. So the approach angle is to relate the variability of the in-hive temperature to the strength of our colonies. And we have two main avenues of analysis in progress for that. The first one is more related to a biological question. That is there a link between the thermoregularity capacities of the colonies and their strengths? The strength is either the number of bees or the size of the brood. And if yes, we want to answer a more practical question, that is to know if we can use this automatic temperature data to inform beekeepers about the strength of their hive. For that, we use for now a dataset from an old project. This project is named ToolBee, and the goal of the project was to test tools for monitoring bee colonies, and the risk of awakening. The data are very similar to Be Good because it is automatic in a hive temperature that is taken in the middle of the hive, as you can see in this schema. We use this sensor at the middle of the hive, and we have one measurement every 5 minutes of a precision of 0.10 degrees Celsius. We also have the 29 colonies that are in Avignon that were evaluated during 2 years. The first years in 2008, we did the project did 6 colony evaluation from July to October, and in 2018, 5 colony evaluation were done from April to July. So to answer our first biological question, we use several predictors. The main predictor that we are really interested in is the colony strength. That is from the colony evaluation. We have 2 possible predictors here, either the number of adult bees or the number of the total number of brood cells that are capped or uncapped. We also added other predictors to because we expected that they can influence the regulation of the temperature inside the hive. So some calendar predictors with the month and year. We also added a the weather predictor. We tested several weather predictors, but we retained only the maximal external temperature that seemed to be the more to have the more influence on the regulation of the in-hive temperature. And also we use the mean temperature that I will describe just a little bit after. We also added a colony random effect to take into account variabilities between colonies. And unfortunately, we could not add interaction because of these predictors, because we did not have enough data, but we might be able to do that with Be Good data later. So to use as explained variable, we use the temperature variation that we calculated with 3 steps. The first step was to calculate the mean temperature over the 24 hours before the colony evaluation. So in our 2 graphs here, you can see the second one is just a zoom of the first one. There is the colony evaluation in red and 24 hours before the blue line. So we use data of temperature between these 24 hours, and we calculated the mean for this period. After that, we also removed data for which the mean was not in the range 30–36 degrees, because we hoped to, by doing that, to remove the cases where the sensor was not near the brood, and because the bees were not taking care of a regulation at this spot. And finally, with this same data of the 24 hours before the colony evaluation, we also calculated the coefficient of variation, that is a measure of the relative dispersion around the mean. We also used as a percentage, as you can see in the calculation here. And with this coefficient of variation, the lower the coefficient is, the lower the dispersion around the mean. So the lower the coefficient is, the temperature is less variable. We did two preliminary graphs here to see if this coefficient of variation could have a link with the strength of the colony. And we can see a link with either the number of adult bees or the brood cell. So we did, to confirm that, we did modelization. So what we did was to add the predictors I talked to you about in a model. So colony strength with the two number of adult bees and brood cell. The month year were one proxy of weather, and the mean temperature as also a colony random effect. We included all that in a model from general linear models, and we tried to explain the temperature dispersion near the brood with that. Because sorry, because our coefficient of variation, that is the explained variable, is positive, continuous, and severely skewed, as you can see in this graph here. We had to use a GLM with inverse Gaussian distribution and not a normal distribution, and with a log links function. Sorry. For the results, we have here tried all the different possible models with all the predictors I presented to you, but no interactions, as I told you. The numeric predictors were all scaled for to make the models easier to make the models easier. Sorry, and we also added or not added the random effects. So as you can see here, we compared the model with the AKK information criterion that is corrected by the number of observations, so AICC. And with this criterion, we selected the best model that you can see here in green. And the best model selected had the number of adult bees as explanatory variable, the months, also the maximum external temperature, and the mean temperature inside the hive, and also the colony random effect. To investigate the results of the model, the best model, here we can see that the more we have adult bees, the less the coefficient variation is important, so the less the temperature is variable. The month of April is taken as intercept, so it is compared to other months. As we can see that all the months between May and October have less temperature variations than the months of April. We also can see that the more the maximal external temperature is important, the more the temperature inside the hive is variable, and the opposite, the more the temperature inside the mean temperature inside the hive is important, the less it is variable. We also can see here the simulation of our model that fits the data with the total number of adult bees, so this predictor here. So that was the first biological question. We still have lots to do on that. We are trying to investigate all better predictors because we want to replace, for example, the months. That is not very interesting for us as a biological question. But once this part will be finished, we want to start the second part, that is the practical question, meaning that we want to try to use the coefficient of variation as a predictor to try to explain the strength of the colony, so that maybe we can help beekeepers to open their hive less often and use the automatic data as a good insight of the hive. We will unfortunately we will have to use other predictors, we think, but this is not work that we started yet because we want to first finish the first question. So to link that with the Be Good project, the idea was to first develop models on the toolB dataset to in order to ask a good hypothesis and to develop properly statistically the models. Then we want to obtain preliminary results, and this preliminary result will be compared to results on the same models on BGood. So the replication of these models on BGood will allow us to compare and to see if we can confirm or not our first results. And we also hope to improve the models by, for example, using interactions, because the BGood dataset will be much more complete. So we will be able to use a more complete model. So thank you for your interest in this subject. If you have any question or even suggestion to improve our thought on this subject, we are more than welcome to hear what you want to say.